All right, so it's 12:02 and we should start. Hello, everyone, and welcome to the grand rounds for the Minnesota Regional Spinal Cord Injury Model System. Thank you all for joining us today. The Minnesota Regional Spinal Cord Injury Model Systems is funded by the National Institute of Disability Independent Living and Rehabilitation Research, NIDILRR, to support innovative projects and research in the delivery, demonstration, and evaluation of medical rehabilitation or vocational and other services to meet the needs of individuals living with spinal cord injury. Next slide, please. The model system represents an exciting new partnership among the following institutions: Carthage County Rehabilitation Institute in Minneapolis, Minnesota. Thank you. The Mayo Clinic, Rochester, Minnesota, Region Hospital at St. Paul, and the University of Minnesota in Minneapolis. Next slide now. Our community collaborators include Minnesota Spinal Cord Injury Association, United to Fight Paralysis, Get Up, Stand Up to Cure Paralysis, Paralyzed Veterans of America, United Spinal Association, and the North American Spinal Cord Injury Consortium. This system collectively provides a multidisciplinary continuum of care for people with spinal cord injury, including emergency medical services, acute care services, acute rehab services, and post-acute services. My name is Ricardo Allina, and I will be the moderator of today's session. I am the Vice Chair of Research and Professor in the Department of Rehabilitation Medicine at the University of Minnesota Medical School, and a part of the model systems. Next slide, please. Just a few housekeeping notes. The University of Minnesota has organized the CME for today's webinar. Please see the chat for information on how to record your attendance and claim CME credit for attending this webinar within 24 hours of the event. This meeting is being recorded and will be posted in the Minnesota SCIMS website. To earn CME credit for today's presentation, please click on the link in the chat. Due to the number of registrants for today's webinar, we have placed phone lines on mute to reduce audio feedback. However, we welcome and encourage audience participation using the chat and Q&A functions. If you have any technical or logistical questions, we ask that you submit those in the chat to Dan Kelly, who will be monitoring the discussion. So we extend a huge thanks to Dan Kelly at the University of Minnesota, who worked very hard behind the scenes to make these webinars possible. Next slide, please. The title of today's presentation is Regionally Specific Neural Progenitor Cells as a Relay for Chronic Spinal Cord Injury. And our speaker for today's webinar is Dr. Parr. Dr. Parr is an associate professor in the Department of Neurosurgery at the University of Minnesota Medical School, as well as a medical director for spine and principal investigator of the Parr lab at the Stem Cells Institute. She's a past chair of both the Minnesota Neurosurgical Society and the Section for Women in Neurosurgery, a joint section of the American Association of Neurological Surgeons. Dr. Parr is a board-certified neurosurgeon, and she is interested in finding new therapies for spinal cord injury. She's also a faculty member in the graduate program in neuroscience and the Stem Cell Institute. Dr. Parr research centers on transplanting neural stem cells grown from a patient's own skin into the injured spinal cord. She has an active translational research laboratory, and she is interested in examining mechanisms of functional recovery using techniques such as histology, immunohistochemistry, cell culture, optogenetics, and animal modeling. Dr. Parr, welcome to our Ground Rounds seminar, and thank you for presenting.
Thank you very much. These are my disclosures. I have no conflicts of interest, and these are the different funding sources for the research that'll be presented today. And thank you for that introduction, and thank you for asking me to speak here today. It's my pleasure, and I enjoy speaking about our laboratory research. So as mentioned, I'm going to be talking about regionally specific neural progenitor cells as a relay for chronic spinal cord injury. I think it's widely recognized in the spinal cord injury community now that there is not going to be a magic bullet for treatment of spinal cord injury. There's not just going to be one treatment, but hopefully there will be many. All spinal cord injuries are different. There's acute versus chronic, cervical versus thoracic, complete versus discomplete. And all of these things are probably going to require different therapies. On top of this, some of the therapies may be combinatorial in nature or involve several different therapies in combination. So this is called the toolbox of therapies. And in our laboratory, we're focused on 3 of these therapies. One is cell transplantation, another is 3D printed scaffolds, and the third is electrical stimulation. And so I'm going to be talking about all 3 of these things today by themselves and in combination. And so the majority of the time is going to be talking about cell transplantation therapy. And hopefully I can kind of take you through our story in our lab of what we've discovered in the last decade of research. I'm going to talk a little bit about the rationale behind cell transplantation, the clinical trials, past and present, our approach in vitro, and our in vivo studies. Then I'm going to talk a little bit about our 3D printed scaffolds and what we've discovered with that. And finally, talk a little bit about electrical stimulation in our rat model. So first of all, transplanted cells in spinal cord injury, how do they work? What's the mechanism of action? Well, I like to categorize things. I think it makes it easier to digest. So I basically categorize the mechanisms into 4 different categories. The first one is what we call neuroprotection. This is protection from further injury, from inflammation and other processes. And neuroprotection this is a little bit debatable, but it's probably only really going to be relevant in acute or subacute spinal cord injury, because in chronic spinal cord injury, many of the secondary injury mechanisms have already passed. But this is basically right after the injury, you give a patient something that prevents further inflammation and further ongoing damage to the spinal cord. The second one is creation of a favorable environment for regeneration. So this is endogenous regeneration. The third one is remyelination, and this involves the mechanism by which, after injury, it is believed that some of the axons that may still be intact across the site of the injury are denuded, or they're missing their insulation, which is called myelin. And by re-insulating those axons, we may get them to work again. So this is the mechanism of remyelination. And finally, there's direct replacement of damaged neurons. And the mechanism behind this is thought to be possibly a relay system, whereby we create a relay across the site of injury and to allow signals to cross in both directions. And so that's mainly the final one here. The replacement of damaged neurons is mainly what we're working on in our laboratory, and that's what I'm going to be focused on today. So it's fundamental to understand when we're talking about cell transplantation and spinal cord injury that all cells are not the same. So different cell types have different potential mechanisms of action. And again, because I like to categorize things, in my mind, there's 2 different cell types. There are neural cells and non-neural cells. So neural cells, as I mentioned, new neurons could integrate into existing neural circuitry, or new oligodendrocytes or Schwann cells. Those are the cells that create myelin or the insulation. They could remyelinate axons. Non-neural cells are the other category of cells. And these are, for example, mesenchymal stromal cells, bone marrow stromal cells. They include OECs, which are also well studied. They include things like adipose-derived cells. And also umbilical cord cells, all these things kind of fall in my mind into the same category. So these cells could provide neuroprotection, or for example, as I spoke, in acute or subacute injury, or they could create a favorable environment for endogenous regeneration. One of the important things to note here, though, is that neural cells can also do those things. They have the potential also to provide neuroprotection or create a favorable environment, but non-neural cells cannot integrate into existing circuitry and create new relays, and they cannot remyelinate axons. So it's important to recognize that. So mostly what I'm going to be talking about today are neural cells. So why neural stem cells? Why all the hype, all the buzz? Well, in the 90s, it was recognized for the first time that stem cells or neural stem cells exist in adult human brain and spinal cord. And this was exciting because for the first time, it was recognized that we have a certain degree of plasticity in adults that we not recognize in the past. And so neural stem cells are very appealing because, first of all, they self-renew, so you can grow them in a dish and make as many as you like. And the second thing is that they give rise to all three CNS cell types, that is oligodendrocytes, astrocytes, and neurons. And so a lot of work was done in the early years looking at these cells, and it was discovered that in amphibians, or specifically urodeles, neural stem cells divide and differentiate to regenerate the spinal cord. So you can cut an amphibian spinal cord and it'll grow back. So in humans, or in mammals specifically, this does not happen. And why not? Well, we found that these cells divide in response to injury, but they have very limited regenerative ability. So it was thought that perhaps we could boost the number of cells and provide some functional improvement. So early studies transplanted stem cells into both small and large animal models and did find some functional benefit. But I will say it was quite spotty, where some people found functional benefit, some people didn't, and the mechanism wasn't truly understood, so nobody was quite sure why or what was going on. And so regardless, this was kind of moved forward into clinical trials in the US, and some would say perhaps it was a bit premature, but we learned a lot from these trials. And so there have been 3 neural stem cell trials, all in the US, that have occurred over the past decade. The first one is called Stem Cells Inc., or the Pathway trial. Their product were fetal-derived neural stem cells. They transplanted these and found they were safe, and then they attempted a phase 2 trial, but this was terminated after an interim analysis revealed that they were not going to be able to be successful in reaching their primary endpoint. And interestingly, there was some debate after the fact about why this didn't work. And it was suggested that it was possible that these cells were not regionally specified. And I'm going to get back to that. And so the second trial is the Asterisk Biotherapeutics, or SCI STAR trial, which is also known as the Geron trial. Their product were embryonic stem cell derived oligodendrocyte progenitor cells. So their goal was to make oligodendrocytes, which make myelin, which can remyelinate the axons, which is one of the mechanisms that I described earlier. They demonstrated safety in a phase 1 trial, and they suggested efficacy, but it was compared to historical controls only, which, as you know, are not terribly robust. So they're planning a phase 2 trial in 2023. They were delayed, I think, because of COVID, but we'll see how. The third group are the neural stem group, and their product are fetal-derived spinal cord neural stem cells. So they're specifically derived from the spinal cord. They did a phase 1 safety trial in ALS and spinal cord injury, and they're currently recruiting at one center, which is UCSD. But so all of these, have been ongoing over the past decade, but none of them have really shown efficacy to date. And so it's time to go back to the bench and to look to see how are these stem cells working? What are the nuances of transplantation? And so in our laboratory, our long-term goal is to initiate a clinical trial using autologous spinal cord neural stem cells to restore and preserve function following spinal cord injury. And I'm going to come back to that autologous point in a moment. So we're interested in using clinical grade human cells that avoid rejection. So that's the autologous part, and integrate into the spinal cord to form a new relay system that can bypass the injury. So again, we're looking at the mechanism of a relay system. And by repairing the spinal cord to create new connections, we believe that we can treat chronic spinal cord injury, which is currently an unmet need. So the first problem that we run into, and this is getting back to that autologous point, is that immune rejection is a real phenomenon. And so this is a really good study in Nature Medicine in 2018 by the Tushinsky group out of California. And this is a bit of a landmark study where they utilized human neural stem cells. They were not iPSC derived. They were, sorry, I'll come back to that in a moment. But they came from embryonic stem, and they created this human neural stem cell line, and they transplanted them into 9 rhesus monkeys. And they injured these, they induced cervical spinal cord injury in these rhesus monkeys. They transplanted the cells. They survived at least 9 months post-injury, and both monkey axons and human axons regenerated into and out of the grafts, and functional recovery was seen. So this was very exciting. But the interesting thing that I found about the paper was in the first 4 out of 9 monkeys, they saw no functional recovery. And when they did the histology, they found that none of the cells survived. So they actually used those 4 monkeys as control animals to compare with the next 5. And so the next 5, how did they get the cells to survive and see functional recovery? Well, what they did is they really bumped up the immunosuppressive drugs, because they found that the monkeys were rejecting the cells. And so after they did this, they found that there was graft survival and fill of the lesion site. But this was quite a high dose of immunosuppressive drugs, which is not optimal for use. So this just highlights the fact that immune rejection is real. And so our solution is to use induced pluripotent stem cells, or iPS cells. And so again, recent human trials, the 3 that I showed you, are using embryonic or fetal stem cells. Again, these are allogeneic, so they all require immune suppression to prevent rejection. And of course, there's some ethical controversy over the use of embryonic or fetal stem cells. So we propose using fully defined autologous cells derived from adult stem cells. So iPSCs, or induced pluripotent stem cells, what they are is we take a patient's skin, and then we utilize certain factors to turn back the clock and turn the cells into embryonic-like stem cells. So then I say like just because then they have the ability to become any of the cells in the human body. So we have the ability to make them autologous, so there's no rejection, because we make them from a patient's own skin. Clinically relevant means that the protocols are also translatable to humans, which means they utilize non-integrating virus and animal-free reagents. So here's just a quick slide demonstrating what iPSCs are. These are human iPSCs. You can see the little person in the top left of this diagram. There's a skin biopsy, and actually you don't have to use skin. You can also use blood or any of the other cells of the body. It's just skin is pretty accessible. Reprogramming factors, these are the YAM and active factors listed here to make iPS cells. And then these cells can self-renew, so we can make more of them. And then in this diagram, you can also do gene correction if you have a disease, underlying disease, but that's not what we're doing here. We're just differentiating them. And then you can also use this for disease modeling in a dish. But what we plan to do is to transplant these transplant these cells back into the patient. So again, these cells are proliferative. They're capable of conversion into all different kinds of cells, and they're ideal for clinical translation because they can be autologous. And again, we are paying attention to using defined cell culture reagents, reproducibility, making protocols that are reasonable, fast, quick, replicable, and regional specification, which I'm going to get back to in a moment. So we ask the question again, what kind of stem cells should we use? Because iPSCs themselves are too primitive. So if we injected those into the spinal cord just by themselves, they'll create tumors by definition. So we need to differentiate the cells, as I mentioned. But the interesting thing is that all stem cells are not the same. And I think this was something that was largely unrecognized in the early days of neural stem cell research and neural stem cell transplantation. And this may explain why a lot of transplantation studies were not successful. So all neural stem cells are not the same. And one thing that has been discovered is that brain cells, for example, do not integrate well in the spinal cord. So therefore, we must find new ways of making spinal cord neural stem cells, or what I'm going to refer to now as SNPCs, but in a fast and efficient manner. And regional specificity is important even within the spinal cord. So not only can you specify it to the spinal cord, you can also specify it to the dorsal, the back of the spinal cord, versus the ventral spinal cord. And ventral spinal cord this is a bit simplified, but the ventral spinal cord, it usually is more motor, and the dorsal side is more sensory. So here's another important paper in Nature Communications a few years ago from the strip matter group, and they used embryonic derived neural stem cells. They specifically patterned them towards the spinal cord. So we know they're patterned towards the spinal cord if they express Hox genes. And they transplanted these into a rodent model. They found that they integrated and produced functional recovery. So that's very promising. So over the past decade, we had really been working on a protocol to make these cells in a fast and efficient manner. And so we developed this a couple of years ago. And basically, this is the paper that we published. And this was Patrick Walsh's work from my lab. And he developed a method of inducing iPS cells in 5 days or less to create all of these different cell types, and including neural crest cells, brain cells, and spinal cord cells. So of course, we're most interested in the spinal cord cells. But one of the interesting things that we did find is that the spinal cord cells that we were making, or what we were going to call the SNPCs, are actually ventrally patterned. So they do tend to be more ventral than dorsal. And so we looked through the protocols to try to find out how we could make more dorsal cells. Because one of the things that's really important in spinal cord injury and is largely ignored is that so many people are focused on motor function. And I think it's partly because it's the most evident thing, it's the most visually obvious thing. But there really is never going to be a cure for spinal cord injury if we don't take into account the sensory function as well. Because even to walk, for example, to walk properly, you're going to need to be able to have sensory feedback of where your foot is in space, and when your foot lays down on the pavement, and so on and so forth. So these are, and for many other reasons as well, the sensory system has largely been overlooked. And so this is my PhD student in my lab, Anne Huntemer-Silvera. This is some of her work, and she went through the literature and looked and found that there are approximately twice as many papers on motor pathology after spinal cord injury than on sensory pathology. So it definitely is an understudied area. And so we've also been working on protocols, and now we have a protocol for deriving dorsal spinal neural progenitor cells. So this is something that we're very interested in. We've been working on for a while. And so we basically use the same protocol. We neurally induce the iPSCs. We cauterize them, which means that we make them into spinal cord neural cells. And then we can either dorsalize or ventralize them after that point to make either dorsal or ventral spinal neurons. And we think this is really important as well. And so this is over the course of 20 days. You can see we induce them after 5 days, but then in order to mature them, it's 20-day cycle. And so then we did some RT-qPCR, and again, this is Anne Huntemer-Silvera's work. We did some expression profiles to confirm that these were indeed dorsalized. And we did find, I'll just briefly skim through this, but we did find that the evidence that they were dorsalized cells. And then this is where we're going with that project. We are now co-culturing those cells with primary afferent neurons, or DRGs, basically nociceptors that are found in the dorsal root ganglia and then project from the periphery into the spinal cord. And so we want to see how those interact with our dorsal SNPCs. And so our preliminary result, and this is just one picture, but we found that the cells, when we co-culture them, they survive, they proliferate, and now we're going to evaluate the effects of co-culture on differentiation and synaptic development. And so this is a kind of a cool picture because you can see that there do appear to be some connections formed between these various cells. So we just have to analyze that and see what exactly is going on. But we're really excited about moving forward with that. So then we wanted to be more translatable. We wanted to see in vivo what happens with these SNPCs. And so for the rest of the project, every time I talk about SNPCs, they're actually the ventral SNPCs or the VSNPCs, because we had just developed the dorsal protocol. We actually didn't have those cells when we started this set of experiments. But moving forward, we're going to be integrating the dorsal ones into our experiments. So this is our first study that we did, and we just published this paper. We started with 2 groups of 10 rats each. These are ATN rats, because even though I mentioned autologous transplantation, these are human cells into rats. So obviously we need some type of immune suppression. So these are athymic nude rats that are congenitally immune suppressed. We had 2 groups, spinal cord injury and spinal cord injury injured plus cells. Pretty simple. We transplanted the cells 7 to 10 days after injury, or a subacute model, and we sacrificed the rats after 12 weeks, and then we processed the tissue and did immunohistochemistry to see what was going on. So this is briefly what we found. So first of all, we found that the cells survived in a very robust way, much more than expected, actually, and we actually wondered if we had injected too many cells. But you can see here in A and C, there's a large clump of green cells in the cavity, filling the cavity. And then we did a cavitation analysis, and indeed, it was statistically significant that these filled the cavity. And then we did immunohistochemistry to find out what these cells became. And interestingly, although these cells are almost 100% showing preliminary neuronal markers, so they look like they're going to all be neurons in a dish, after we transplant them, they actually become all the different cell types of the spinal cord, which we found very interesting. And we found that repeatedly now in different experiments. And so about half of them became neurons, and we looked at 4 weeks and 12 weeks as well. But we found that at 4 weeks, most of them had already differentiated. So there wasn't a huge difference between 4 and 12 weeks. But basically, about half of them became neurons, about a third of them became oligodendrocytes. Some of them remained in a sort of a pre-differentiated state. Very few, like 5%, became astrocytes, and then there was less than 5% proliferation. And so we found that very interesting. And then we wanted to confirm this by doing RNA-seq. And so we used laser microdissection to cut out the cells in that transplant area. And we compared them to the cells in culture before transplantation to find out what differences were there. And so we found that compared to the ones in culture, they expressed increased Hox genes, which shows regional specification again. They showed more neuronal genes, gene expression, but more differentiated neurons. And then ionic channel genes and synaptic genes, demonstrating that these were more differentiated towards the neurons were more differentiated. Sorry. And finally, we looked at where the axons went and where they were extending to. And we found that the graphs extended long distances, even up into the brain, which was a little surprising, but it has been reported by one other group. And we found, interestingly, that if gaps existed between the graft and the host tissue, no axons extended. So that's shown in figure D here. And we also found the presence of both MBP, which is myelin, and synaptophysin in close proximity to the graft. So this suggests both myelination and synapse formation. And so we found that really interesting. Unfortunately, in this study, we didn't get any functional recovery, and it may be the study was underpowered, but we had some very interesting findings, and that was sort of our first preliminary study with the cells. And so we have then asked ourselves, so now I'm going to talk a little bit about our 3D printed scaffold work. So we asked ourselves, though, is there a better way of delivering these cells rather than just sort of squirting them in and having them go everywhere? Can we place the cells more precisely? And can we avoid this gap between the cells and the host? And so that's when we decided to get into some 3D bioprinting work. And we did this with Dr. Michael McAlpine, who is in the Department of Engineering. And basically, we developed a method of printing the scaffolds and then printing the cells directly into the scaffolds at the time of printing. And so A just shows the scale at which we can print scaffolds. And then B shows, and so all of this is in vitro. So this is all in a dish. And so B, C, and C just basically show when we printed our SNPCs into the channels that they form these really robust axonal networks. And I'll get back to D in a moment, but basically E and F are demonstrating that one of the advantages of 3D printing is that you can print something in any shape you want it to be. So the eventual idea is that we can do an MRI of a patient's spinal cord injury. We can then use computer-aided design to actually print that shape in a scaffold shape that we could then transplant into the site of injury. And here, this pretty picture at the bottom is, again, just using immunohistochemistry to demonstrate one of the channels. And again, the green are the extensive neuronal networks that form. And here, yeah, okay. And so we also wanted to try to co-culture 2 different cell types to see whether we could also specifically place different cells in different areas. And this will probably become relevant later on when we start printing the ventral and dorsal SNPCs, because we can print them in the ventral area or the dorsal area. And this is something that we're planning to do in the future. But basically in this experiment, we printed both the neurons and the oligodendrocytes together. And we found that we could print them together at the scale required, that they both survived. And the neurons that were here were functionally active. So we used calcium imaging to demonstrate that they were. And we also did some patch clamping on them. And we found that they formed functional neural networks more quickly than traditional 2D culture when we compared it to the 2D neurons in a dish. And we also found that the oligodendrocytes can form myelin, and that's in C. The red is myelin. And so the other interesting thing we found when we printed these in the 3D construct is that they actually form these organoids or mini spinal cords. And so we had noticed before, as I mentioned, when we take these SNPCs and transplant them into a spinal cord, even without the scaffolds, the spinal cord itself is also sort of a 3D, it's a 3D construct. So we found that the cells, though, really don't just stay as neurons. They form different cell types of the spinal cord. We thought this was curious, but in the spinal cord, because when you print them, they kind of go everywhere, they weren't really forming a definite structure. They were just sort of randomly forming these different clusters of cells. Whereas when we put them in the scaffold, we found that, interestingly, they started layering out. And by that, I mean that the natural spinal cord has specific layers to it. And in the ventral spinal cord, the layers 1 to 3 express FOXD3, CHEX10, CHEX2.2. And we found, and this is what this diagram is showing, those 3 markers in subsequent layers. So we were really excited about that. And again, we found that in 3D culture, they matured faster and formed enhanced neural networks. So we thought that rather than like throwing spaghetti at a wall by transplanting the cells kind of willy-nilly, we're actually making lasagna, where these are developing specific layers that are recapitulating the development of this natural spinal cord. So we were very excited about that, and we went on to do some RNA-seq on those organoids as well, to see whether they really were mini spinal cords, and we compared them to the 2D culture of the cells at the same time points. And we did find that they had increasing regionalization, neural development, organoid formation, and post-mitotic neuronal expression. So they were maturing faster, and they were regionalizing, and basically they were forming these mini spinal cords. So we were pretty excited about that. So this is an ongoing project. So the next thing we did, as you might have guessed, is we transplanted these scaffolds into several groups of rats. And so that tissue is still under analysis. So I don't have those results for you today, but I do have the locomotor function testing, because we have finished those experiments. And we did find, which is exciting for us, that we had 3 groups. We had the control, which had no scaffolds. We had a scaffold only, and then we had the scaffolds with the cells. We found that the scaffold only did show a slight improvement in function. And these are transected animals, so they really don't have a joining spinal cord at either end. And these are acute as well. So we cut the spinal cord and put the scaffold in, and that was just for the sake of simplicity to make sure we could actually do it and to see kind of what happened with that. But we did find that there was a statistically significant improvement in BBB scores with the scaffolds with the cells in them. So we're pretty excited about that. But again, we have to analyze the tissue and to see really what's going on. So the final thing I'm going to touch on today is electrical stimulation. So this is the third sort of leg of the stool for us. We want to also add this in. And so we have a model of electrical stimulation in a rat model, which involves, we call it TANES, or tail nerve electrical stimulation. So luckily for us, animals with tails, their tail is actually an extension of their spinal cord. So by stimulating the tail, you can actually stimulate the spinal cord. So it's a non-invasive, relatively straightforward way of applying electrical stimulation to rats. And so the reason that we thought this was interesting is because we also asked ourselves the question, we're putting these cells in there. We've shown that they are able to form some connections with the native spinal cord, and the native spinal cord is able to form connections with them. So that's kind of the first step to understanding how a relay system works and being able to show, yes, it can form a relay, but how do you know it's a useful relay? And how do you know these are correct connections and not aberrant connections? And secondly, can we train this new relay system to function more optimally? And again, this would be recapitulating development, because in development, our new brains make all kinds of different connections, and some are useful and some aren't. So how do we know, or how do our brains develop in a useful way? Well, basically, the connections that we use all the time are reinforced, and the ones that aren't are pruned away. So can we take advantage of this neuroplasticity in the spinal cord by providing this electrical stimulation in order to tell these cells where they should connect? So this is a study, again, that we're sort of wrapping up right now, but this is our most recent data, sort of hot off the press, but this is unpublished as well. So we did this study that is called Training Transplanted SNPCs to Function After Chronic SCI. So in this study, we took 3 groups of 10 rats each. We had spinal cord injury only, spinal cord injury plus cells, and then spinal cord injury plus cells plus TANES, the electrical stimulation. We transplanted the cells 8 weeks after injury. So these were chronic rats. So we injured them and left them alone for 8 weeks. And then we started our study. After that point, we did daily TANES and weekly locomotor testing, so BBB scoring. We sacrificed the rats at 16 weeks, and we processed and examined the tissue, and the tissue examination is still ongoing, but I have some results here. So this is in vitro, and again, as I mentioned before, in vitro, all of our cells express beta-tubulin and NF200, which are neuronal markers, but no expression of oligodendrocytic or astrocytic markers. So we transplanted these cells, and these are, I won't show the control group because there's no cells there to see, but this is basically the cells only, or the cells plus TANES. And these are sagittal sections. And we can see that we have definite SC121, the green, it marks for the human cells. So you can definitely see that we have survival of the cells. There's a slightly different pattern here, but it's definitely survival, and NF200 is neuronal marker. So we know that they survive, and we know that with cells and TANES that they both do differentiate somewhat into neurons, although I don't have the numbers yet for that. And then when we looked at the differentiation profile, we did find that they produced, and again, we haven't done all the cell counts yet, but I can tell you that they do produce some astrocytes, some oligodendrocytes, and mostly neurons, but just not sure the proportion yet. But in all cases, though, there were more of all the cell types in the TANES animals than the cells only. So that was quite interesting. And I'll come back to that in a sec. So we also looked rostral and caudal to the lesion. So with the cells, these are axial section, 6 millimeters rostral, and then 6 millimeters caudal. The rostral's on the left, the caudal's on the right. And so it's a little bit difficult to say without cell counts, but it does seem pretty obvious to us when looking at the cells that there were more axons in the TANES group than in the non-TANES group, rostral and caudal to the lesion, which was very interesting. And so again, we also looked at astrocytes, oligodendrocytes, and neurons, rostral and caudal to the lesion. So overall, we found that the cells with the TANEs resulted in higher survival of the transplanted cells than compared to the cells alone. And this is so far, again, observational, but we believe that TANEs results in higher cell survival. And just within the cell group itself, we found that more axons and myelin were seen caudally than rostrally, which is sort of an interesting thing, but we're not quite sure what that means. What I think is the most interesting finding so far is that both axons and myelin were increased by the TANEs rostrally and caudally. And it was pretty obvious observationally that this was occurring. So whether that's a function just of cell survival, because if there's more cells surviving, then there's going to be more axons and myelin, or whether there's another mechanism remains to be seen. But this is very interesting. And also given the fact that one thing we know is that axons are more likely to be myelinated because when they're active. So an active axon will actually act as an attractant to myelinating cells, the oligodendrocytes, and they're more likely to be myelinated. So we're wondering whether by stimulating these remaining axons, they're causing them to be myelinated more robustly. So finally, we have our BBB scores, or locomotive scores from these from this experiment. And again, I will point out that to emphasize the differences, I have expanded the BBB scores on the left-hand side from 10 to 15, but in actual fact, it goes from 0 to 21. These are contused animals. So at baseline, which is the green, it's saline only, we would expect them to be pretty stable. And so they're about a an 11 or 12 on the scale, maybe 11. And then the cells only, we noticed a slight trend towards improvement, but it wasn't statistically significant. And then with the TANEs, we noticed an obvious improvement right from the beginning. And these animals, unfortunately, it's not statistically significant. We think that the study is underpowered, and in the future, we're going to use more animals to try to sort of suss this out. But the interesting thing we noticed about the trend too is that I can't say for sure that even after 16 weeks that the TANEs isn't going to continue to improve the rats who are receiving it. So I found that interesting as well, and lots to think about. So in summary, what we talked about today, I talked about considerations of transplantation therapy, the importance of the immune response and the use of iPS cells to deal with that, and the importance of regional specificity. And specifically, I called out the role of dorsal cells and the underrepresentation of sensory function in the spinal cord injury literature. Then we talked about the potential for 3D bioprinted organoid scaffolds, which is ongoing research. And finally, the need to understand the mechanisms of action of electrical stimulation. And I think that, as many of you know, we have an ongoing clinical trial in humans looking at the role of epidural stimulation. But there really needs to be more understood about the mechanisms of action, I think, in order to optimize that therapy. And future plans, our next study is going to be combining all three things in a combinatorial therapy. Our cells, our SNPCs, which include both our dorsal and ventral cells, our 3D scaffolds, and our TANEs. And I'd like to acknowledge all the people in our lab that contributed to this, and also in the McAlpine lab. Our collaborators, including Mike McAlpine, who does the 3D bioprinting, Eric Holmberg, who's consultant on the TANEs, James Dutton, who has been working with me on the cell protocols over the years, Walt Lowe, who's a transplantation expert, Manny Esguerra, who did our electrophysiology and the patch clamping for us, a list of our past funding and our current funding. And then if anyone has any questions or anything after this is over, you're also welcome to email me, and this is my email address. And thank you again for allowing me to speak here today.
Well, thank you for an excellent presentation. And I see there's a few questions in the Q&A box. I have a few questions myself. But before we start that, I would like to introduce Matthew Roderick, our discussant for today. Matthew is father to his son, Gabriel, who sustained a C5 complete injury in 2008. Matthew was his son's primary caregiver for a number of years until Gabriel moved on to live independently. Matthew then turned his energy to advocacy, first in initiating and leading the effort to pass the Minnesota SCI-TBI Research Grant Act, co-founding Get Up and Stand Up to Cure Paralysis, and then the opportunity to lead United to Fight Paralysis as executive director since 2017. Matthew serves on the Minnesota SCI-TBI Research Advisory Council, the Minnesota SCIMS Community Advisory Committee, Get Up and Stand Up to Cure Paralysis Board, and was a former executive member of the North American SCI Consortium. So Matthew, welcome and thank you for being here.
Thank you, Ricardo. Very honored to be the discussant today. And thank you, Dr. Parr, for the presentation. Interesting stuff. As Ricardo said, I'm father to a son with a spinal cord injury, so I take a great interest in this area of research, particularly in the area of regeneration. So Anna, have a bunch of questions for you, but I know I have to keep them short and maybe consolidated, so I'm going to kind of compress them a little bit. The first one, for folks in attendance with a spinal cord injury from the community, and I'd like to focus a little more on both the cells and then your ultimate plan to combine them. What's the big takeaway from what you've just described to someone living with a spinal cord injury, especially, given the fact that there are folks, including you and others, who have done some of this work and shown that cells will differentiate, cells will survive, cells will connect, but there's very limited to no function. And so where is this going? And what even, in a sense, can you hypothesize about why is that? You and your colleagues have learned a great deal about the cells, about as you described with the lasagna metaphor, which I liked, the different layers that are required, the architecture that's required. So what's missing? Is it a disruption that's necessary to sort of disrupt that chronic state? Is it a more biologically active architecture? If you can answer that question, again, what the big takeaway is, and kind of what you can hypothesize about why is there's all this survivability and connection, but no function.
Yeah, that's a great point. I think the best way to describe it, what I guess I've come to, realize is that it's very rare in science that we have a huge leap from one thing to all of a sudden we have a leap to a cure or a treatment for something. And in history, we can probably count on one or two, or both hands how many times that's happened. And a lot of the time, unfortunately, it's baby steps, and it feels like we're inching along. And I understand that it's really difficult and frustrating. And I think it's frustrating obviously for the spinal cord injured community, and it's frustrating for families, and it's frustrating for us as providers to not be able to say, okay, we've made this advance forward. But I think that if you take things in the context of like the last 20 years, I really think that we have made a huge step over that 20 years. When I look back to when I started research and I look at how poorly understood neural stem cells were, I didn't even know before the 90s that adult neural stem cells existed. And then we understood it and we were managing to grow them, but we were sort of all lumping the stem cells into one clump. We were just saying, oh, the neural stem cell is a neural stem cell, but that is so false. That's so false. And so I think that while we understand things so much better now, I do think there's a few tweaks, and some of them are technical to get this to the next stage. I mean, even like I say, the Pathway clinical trial, which I know you're very familiar with, that's the stem cells England. I was a PI on that, a site PI on that one. And I injected people with stem cells. And unfortunately, it didn't really do a whole lot. It was safe, but it didn't really do a whole lot functionally. But there was a lot of criticism after that trial that those cells were not regionally specified. There were a lot of people that believed that those were actually brain cells that they were putting into the spinal cord. And now we know that brain cells don't work. So if we had maybe had that knowledge back then, maybe things would have, turned out a little bit differently. So I think that there are these important steps being made. And I think that functional recovery is possible. And I do want that to be a takeaway message from this. I don't think it's just that we've shown all this connectivity and there's no functional recovery. I think that there are studies that have shown that functional recovery is possible. And one of those is the Tushinsky study that I showed. And there are a couple of others, the other one that I showed from the Strip Matter group. But I think that a lot of us aren't getting enough connectivity, for one thing. So is it that we're putting in too many cells? Is it that we're not putting in quite enough cells? The other thing that's come up recently is that are we not, while we're regionally specifying to the spinal cord and we're regionally specifying dorsal or ventral, are we not specifying enough for cervical, for upper or lower cervical, for upper or lower thoracic, for upper lumbar? And as we do, even though some of this basic science research looking into dorsalization may seem kind of peripheral and irrelevant and to the clinical patient, I think it's actually very relevant, because I think these are the types of things, these little nuances now that we're getting down to. And I think it's also really promising that we are able now to get down to these nuances and to ask these questions about these tiny little things to say, is this better or is this better? Is this better or is this better?
Thanks. Relatedly, back to your long-term goal, of going to deliver a therapeutic, taking a therapeutic to trial, or a combination of. And your very last slide of your next steps to do cells, scaffold, and stimulation. I'm wondering if you can just sort of comment on, the way people talk about combinatorial therapies now and the challenges because of the regulatory pathway and the idea of bringing 2 or 3, 4 therapeutics together is really a challenging proposition. Can you see a future in which those things are actually staged clinically? So hypothetically, spinal stimulation is being studied in human beings right now. Maybe that comes over the threshold of approval and becomes clinical practice. Now you've got people with spinal cord injury that have a piece of what you're proposing to combine. And then subsequently in the real world, they can add therapeutics to that from the community, recognizing that spinal stimulation isn't a cure, but maybe in combination with other treatments or therapeutics that might be more curative, more restorative in nature. So how do you see that unfolding? And relatedly, the question also I wanted to ask is, what for you is the sort of go decision? What do you need to see from your studies going forward to make that decision to pull the trigger on your long-term goal? Take this out of your lab and try to take it to human beings. What needs to happen for that to take place?
So I think that probably my answer isn't really going to be an answer. It's just going to be adding to what you say, because I think all the things you said are very important and true, especially about combinatorial therapies. I think it's really important to always be working in parallel tracks on each thing separately. And that's kind of what I tried to show here. Is I tried to show those things together, separate, and then sort of together to a certain degree. So just showing like the logical progression of having all these parallel tracks and kind of combining. So as you know, we're working on the epidural stimulation in humans. We're working towards expanding our clinical trials. We're looking at trying to get regulatory approval, or actually FDA approval, for this to be more widely available to other people. So that's one track we're working on, which, I haven't presented anything about that today, but believe me, it consumes a lot of my time. And, and then on the other hand, we're working on so the scaffolds are also being worked on by people other than myself, not our 3D printed scaffolds, but there is, as you know, in vivo therapeutics have been doing this trial where they're implanting scaffolds into humans. And that's really the first step, because they have approval for their scaffolds in humans. That's a huge thing in my mind. And that kind of leads the way for the rest of us to be able to pursue this kind of work. And I think with the cells, it's always going to be a lot trickier. But I think that Japan has kind of led the way in this field with special approval for iPS cell technology. So they're the first ones that are allowing this in humans. And I think that based upon what they find too, that will be kind of guiding, I think, towards a little bit for the US FDA to sort of make some considerations about this. So I think that working on all these things sort of separately. And as for sort of the go thing about, how can we move this forward to the next stage? I think that there's a couple of things. I think, eventually, and we've talked about this a little bit before, if we really want to move this forward, we're probably going to have to find a company that to partner with, an industry to partner with. Because I think that sometimes it just becomes too much, because a lot of the regulatory things, scientists and clinicians like myself are not the experts on that sort of thing. So we need somebody who is. And I think, I think that's one of the main things. The other thing is that it would be nice to be able to try some of these things in a larger animal model. And, we have discussed this with a few different people, whether to develop our own animal model or whether to go in a different direction and use somebody else's established animal model. So that's some of the things that are ongoing. So there are always, like I say, baby steps, but there's always like 10,000 different things that we're doing to kind of move each aspect of this forwards.
So, Dr. Parr, in the interest of time, we have only a few minutes to go, and there's a bunch of questions.
Okay.
If you could briefly address a couple of them. Somebody is asking, what makes a clinical grade human cell a clinical grade human cell? And somebody else is asking me, what are the most promising treatments today? So if you could briefly explain us, what will it take for a human stem cell to be of clinical grade, and what are the promising? And then we'll see if we have more time for more questions.
So I'll try to be brief. So a clinical grade human cell is, there has to be certain criteria that are met. One of the criteria is that there is no potential for, because we use viruses to make an iPS cell, we have to make sure that those viruses are not dangerous to humans and that they're non-integrating, so they don't integrate into your own DNA. And so that's one criteria. And so there are viruses like that, but we have to use specific ones. And then another thing is that we can't use animal products that are, could be disease carrying. Another thing is that we have to know what all of the components are. So for example, there's something called Matrigel that we commonly use to grow cells on, but it's a proprietary thing. And because it's owned by a company, they don't tell you exactly what's in it. And so you would never be able to use that, because if you don't know what's in it, then, how do you know if it's safe? And the final thing is that it actually has to be made. The cells have to be, technically, they can't be made in a lab. They actually have to be made in a special facility, which is called a GMP, or Good Manufacturing Practice facility. So those are all things that we also have looked into in the past. We currently don't use a GMP facility, but we have one here at the university that we've had talks with before about generating these cells. And then with respect, I see Rob has asked, what is the most promising treatments today? I would say, honestly, just because it's already out there, I would say that epidural stimulation is going to be the first thing, because it's kind of already out there and it's available to a lot of people. And we're looking at expanding those trials to humans. And I think that the reason that that is sort of fast tracked is because of the fact that epidural stimulation was already being used as for another purpose. So it's always easier to use something that's already being used. If there are, for how long until clinical trials, it's hard to know, but it also, because what I just said, that if things are approved in one area, then it's easier to get them approved in another area. If it happens that iPS cells do become approved, for example, for acute macular degeneration, which does look like a pretty good indication for them, that's eye problems, then it may be easier then to move it into the spinal cord injury realm. Or if it shows great success in Japan with their studies, then it might become easier and then it might move along more quickly. But it's a little hard to say with, yeah, it's a little hard to say exactly a timeline.
I had a question myself. I was always a fool for developmental biology, and I hadn't appreciated until today this aspect of dorsal versus ventral. And so when you do the process of caudalization, I was also wondering whether, as we become more familiar with the system and we learn more, if it will be important what type of cell you use depending on where the injury site is. Because, you know, as you know, Hox expression determines patterning, position, and this is something that maybe we still are addressing in a way that is not fully clear to us, right? I mean, that this is everything when it comes to regeneration of tissue has to do with location. And even within the core, you know, two different neurons may behave differently even if they are separated by a few segments. That's just a start, and I'm sure you're very aware of this. I have finally one more question. Somebody is asking, how do you know that you're actually regenerating the cortical spinal tract, and that your BBB results are not a result of a compensatory mechanism that occurs below the site of injury versus recovery of voluntary locomotion?
Yeah, so right now we don't know, because we haven't finished both the BBB scores that I showed you. We haven't finished the analysis of the tissue, so I can't say that it's not a compensatory mechanism. We're certainly looking at all the tissue below the cord and everything to see of what we can see, whether there's differences. But in the future, we're actually going to use optogenetics to label the corticospinal tract and then stimulate it, see whether we're getting connections in the graft site. So we have some experiments planned in the future to look at that. We can also use, track tracing and things like that I'm familiar with. We haven't used that to date. We're kind of, again, sort of one step at a time, doing these preliminary studies to say, hey, what's happening? What do we think is happening? And then in future studies, based upon what we see here, if we really want to look at corticospinal tract regeneration, which again is really important, we're going to use some optogenetics to do that.
Okay, well, again, thank you. We are 5 minutes over time. It was really a fascinating seminar and very thought-provoking, and it, personally, it made me think about a lot of stuff that I hadn't thought about recently. And so thank you all for I believe that there is one last slide there. Thank you all for attending today's Grand Rounds. The recording for the webinar will be posted soon in our website. As a reminder, please see the chat for information on how to obtain CMA credit for attending today's webinar. And at the end of the webinar, a brief 3-question survey will pop up. We would really appreciate if you complete this survey, and so we can get your feedback. We also hope you can join us for our next Grand Rounds on Thursday, April the 7th at noon, where Dr. Anne van de Winkel will present on the effects of cognitive multisensory rehab on neuropathic pain and sensory motor function in adults with spinal cord injury. Thank you all, and have a great rest of the day.